Alright, so this is the Chapter 6, Segment 4 outline. In the very beginning of this segment is the last part of the visual system, since this chapter is about sensation. So at the end of the chapter, we're talking about sensory systems. So this is a continuation of the visual system organizes perceptions of groups and objects. And real quickly reviewing what it's talking about, it's referring to Gestalt psychology, which is a German school of psychology that emphasizes that the whole perception is more than just the sum of separate sensations. And there are several rules of perception regarding vision, and some of these are the rule of continuity, which means that we tend to impart continuity, assuming that objects are permanent. So the example the book gives, if there is spikes that seem to be in a circular form, we will see a circle that forms them even though a circle is not there. The rule of proximity, that is that we have a tendency to perceive objects that are close to one another as a part of a whole. There's the rule of similarity, which is the more figures resemble each other in color, texture, shape, other characteristics, the more likely we are to regard them as part of some whole. There's the rule of closure, and this is our perception. It tends to fill in gaps, closing them off to perceive a whole object, even when there might be breaks in lines. And there's the rule of symmetry, which is we perceive symmetrical lines as being part of a single object. And those are all different Gestalt rules of perception. Now, in this segment, the first sensory system that really we take a deep dive into is hearing and the auditory system. And the first thing we talk about is sound is a pattern of pressure in the air or water. So sound can be understood as a result of a pattern of molecules bumping into each other. So air molecules in motion bump against each other, resulting in alternating waves of high and low density called sound waves. When these alternating waves of pressure hit our ears, we hear sound. So the example that the book uses to kind of compare how sound bumps into air particles and moves the molecules into our ears so that we detect this sound is, if you think about it, let's imagine a group of people lined up and they're at a movie theater. And it's a long line, and if someone at the end of the line was to push the person waiting in front of them, that person would bump into the person in front of them, and this bump would continue up the line. The initial person's push might affect people at the other end of the line, and likewise, air molecules in motion bump against each other, resulting in alternating waves of high and low density called sound waves, which I had already said. If the waves of pressure are close together, we experience the sound as high pitch. If we experience low frequency pressure waves, as low in pitch. So the next part that we talk about is the actual structure of our auditory system in our ear, and the outer and middle ears funnel sound to the cochlea. So the first job of the auditory system is to capture sound waves so that they can be analyzed. So looking at the structures of the ear, the outer ear it's also called the pinna. It is a visible part of the ear and the canal leading to the eardrum. And the middle ear is comprised of the eardrum and 3 tiny bones for conducting sound. And the 3 tiny bones in the middle ear are the ossicles, and these help amplify sounds. Then the inner ear is the tiny cochlea, which has a snail-type shape, and some similar organs that are important for balance. And the cochlea is a bony, fluid-filled, spiral-shaped tunnel of the inner ear that detects the vibrations transmitted from the tiny bones, the ossicles, in the middle ear. And finally, the oval window is the opening from the middle ear to the cochlea of the inner ear. So how this all works is the cochlea detects the various frequencies of sound. So the easiest way and how the books puts it to understand how the cochlea works, it can sometimes help to imagine if we unwound the cochlea from its snail shape into a long straight tube. So we have this long straight tube that at one end of the cochlea is the oval window where the ossicle called the stapes from the middle ear passes on vibrations from the eardrum. The vibrations from the stapes push on the oval window, sending vibrations into the fluid inside the cochlea. Remember that the cochlea is fluid filled. So high frequencies displace the membrane and the base of the cochlea, and low frequencies display the membrane and the apex of the cochlea. So the base is closest to the oval window, and the apex is at the farthest point away from the oval window. So the vibrations of the oval window cause vibrations in the fluid of the cochlea that displaces the membrane. So if two different frequencies, one high and one low, they're transmitted together, then two different parts of the membrane vibrate, which I just talked about. So one near the base, one near the apex. So also the louder the sound, the stronger the vibrations, and therefore the greater the wobbles in the membrane. So how specifically that this works and it's translated to sound is the cochlea picks up good vibrations and bad. The cochlea contains several rows of specialized receptor cells, each of these which has a little hair-like extension pointing up away from one of those membranes. And these are typically called hair cells, and these are what the specialized receptor cells are called. And as the membrane vibrates up and down, the upper membrane slightly bends the hairs of the hair cells. This produces an action potential in the neuron underneath the cell, and which travels along the auditory nerve until it enters the brainstem, which then the vibrations is interpreted as sound. Now, we know now how sound is interpreted and how our auditory system works, but how do we know where a sound is coming from? And this skill is called sound localization. It's the ability to perceive the source of a sound. So basically how your body does this is through binaural two-ear cues, which is information about the location of sound found by comparing input to the two ears. So it takes both ears to figure out. One binaural cue is the intensity of difference, and it's a comparison of the intensity of the sound that reaches the two ears. So, for example, if I hear a sudden bang and it is louder on my left ear than my right, then the source of the sound is probably somewhere on my left. Then the book also discusses the duplex theory, which is the idea that both intensity differences and latency differences are used to localize sound. So I had just talked about intensity differences. So latency differences refers to the different times a sound arrives at the two ears. So if a sound arrives at my left ear before the right, the source of the sound is probably on the left. So the duplex theory is saying that it's not either intensity or latency, but probably a combination of the both. The last sensory system we talk about in this chapter is taste and smell, or olfaction and taste systems. And our nose and tongue are exquisitely sensitive chemical detectors, and these two systems are paired together because they have they're not as independent from each other as people may think. Whenever we eat, they're both actively responding because chewing food not only exposes your tongue to the chemicals, but it also generates odors that reaches your nose. So the flavor of food, or the taste, is the combined stimulation provided to taste receptors in the mouth and olfactory receptors in the nose while eating something. So first, the 5 taste receptors contribute to flavor. So the first thing or aspect that we talk about in the taste system is taste buds, which is a collection of 50 to 150 cells, including taste receptor cells and support cells on the surface of the tongue, back of the mouth, and roof of the mouth. Next is a taste pore, which is an opening in a taste bud on the surface of the tongue where chemicals in the mouth encounter the surface of taste receptor cells. So the actual taste receptor cells are these cells on the surface of the tongue that produce taste receptor proteins that actually detect one of the 5 tastes. And a taste scent is simply a chemical that encounters the taste receptor cell and excites it. So the different types of tastes, they're pretty commonly known, but they're triggered by different things. So sweet is detected by, all of these tastes are detected by taste receptor proteins, but there are taste receptor proteins that respond to specific things. So, for instance, sweet, it is proteins that respond to sugar molecules. Sour, proteins that allow positively charged hydrogen ions to enter the cell. The taste of salty is detected by proteins that allow positively charged sodium ions to enter the cell. Bitter is detected by proteins that respond to a wide range of chemicals. And umami, which is a Japanese word for meaty, and it's also referred to as savory, is detected by proteins that respond to amino acids. The last specific things that are talked about in this chapter are different aspects of olfaction, and thousands of different odor receptors bring us the richness of flavor. So olfactory receptor neurons are sensory neurons that detect odors and are found embedded in the sheet of cells lining the nose. The olfactory epithelium is the sheet of cells lining the inside of the nose in which the olfactory receptor neurons are embedded. So the olfactory receptor neurons are in the olfactory epithelium. Odorants are molecules that can be smelled, and the olfactory bulb is the part of the brain that receives impulses from the axons of the olfactory receptor neurons.